Well, here we are, chapter 16 and 19. We're going to talk about the nucleus and finishing up talking about proteins, how they're processed, and how they get to where they want to go. So, let's start talking about the nucleus. So these are electron micrographs. I'm going to see, sorry if I can get, so here, outlined in yellow, if you can kind of see that, that's the nucleus. Right? And that's the same thing shown over here, obviously slightly different nucleus. We'll talk in the next slide about the different structural features, but remember that a nucleus is only found in eukaryotic cells. So prokaryotes do not have nuclei. They have their DNA just hanging out inside the cytoplasm. But eukaryotes have this double-membraned organelle that's full of DNA or chromosomes. And so one of the things I want you to remember is that a chromosome is made up of DNA and proteins. And the proteins are called histones. Okay, so the histones actually help organize the DNA so that it doesn't all get tangled up, like if you've ever had a ball of yarn that's come unwound, or necklaces in your jewelry box, they become tangled up very quickly, so you want to keep them organized. The other part of the nucleus, whoops, right here, that dark spot, yikes, is called the nucleolus, and that's shown here. And we'll talk about what happens there. The nucleolus is important to remember. It's just an area in the nucleus. So it's not membrane bound. It can move around. Cells sometimes have multiple nucleoli in the nucleus. We will talk about the nuclear pores. And euchromatin and heterochromatin, you'll learn about that in genetics. That's different types of chromatin based on how tightly they're compacted. So if I'm asking you to identify structures in a cell, I want you to be able to recognize the nucleus, and if there's a dark area in the nucleus, that will be the nucleolus. I won't ask you to identify heterochromatin, euchromatin, that kind of stuff. All right, so this picture is a schematic showing more information about the structure of the nucleus. So again, what's most important to remember is that the nuclear envelope is made up of two membranes. So most of our organelles only have one membrane, except for those that we believe have come into the cell through the theory of endosymbiosis, like the mitochondria and the chloroplast. Those are also double-membraned organelles. Okay, so there's an outer nuclear membrane and an inner nuclear membrane. The outer nuclear membrane, I want you to see here, is continuous with the ER. Okay, so last chapter in your chapter objectives, I wrote a note saying, I consider the outer nuclear membrane or the nuclear envelope in general as part of the endomembrane system because you can see it's continuous with the ER, which we know is a major component of the endomembrane system. You can also see all these green dots. So there are ribosomes that are attached to the outer nuclear membrane. I've done a bit of research and I cannot find any data that says these ribosomes on the nuclear envelope are actually involved in transcription. So maybe they're just hanging out there, maybe as these membranes move around. But we're going to talk about, or I think we did, is that translation always starts in the cytoplasm, and then if it's going to be part of the endomembrane system, moves on to the rough ER. So these ribosomes are likely just hanging out there. Again, here's that nucleolus region, right, space. Chromatin, we call them chromosomes when they're nice and condensed, when the cell's getting ready to replicate. When they're kind of loose and all scattered around the nucleus like this, we call them chromatin. I'll show you a picture in a little bit, but the nuclear lamina is right along the inside of the inner membrane, right? So this is giving the structure to the nucleus. And then we've got lots of these pink little things, which are your nuclear pores, and we're going to talk a lot about those. When we get into Unit 3, we'll talk about the cytoskeleton, and you'll learn that there are cytoskeleton fibers that attach to the nucleus to hold it in place in the cell. Okay, nuclear pores. This is the way things get in and out of the nucleus. So we've talked about active transport and facilitated diffusion. So this is kind of examples of that, but very specific to the nucleus. This pore has to cross both membranes. This is an electron micrograph showing you these kind of flower-looking things. Those are the pores. They're made up of these complexes of proteins. It's estimated about 30 different proteins. And again, the nuclear pore controls what goes in and out of the nucleus. So just to zoom in a little bit, the book talks about there's two types of transport through the nuclear pore, right? There is simple diffusion, okay. And this is for things like ions, where am I going? NTPs, so these are our nucleotide triphosphate, so like ATP, GTP, TTP, UTP, CTP. So those nucleotide subunits that make up RNA and DNA, and even some small proteins like the histones can go through this pore. And so what I want you to see is that this pore has this outer ring, and then you can kind of see on the side of this purple part in the middle, which is called the transporter. We'll talk about that in just a minute. Okay, things can, if they're small enough, diffuse in and out, right? The fusion's based on concentration, so they're going to go from high to low, and they can go in either direction. So you can imagine that if you have a histone protein out here, okay, and the rest of the proteins are bound up in the DNA chromatin, then the histone proteins could go right in, and this is really important during DNA replication. All right, so let me get rid of this. The other type of transport through the nuclear pore is active transport. And you know that when we say active transport, that means it requires energy. So we'll talk about using actually GTP sometimes instead of ATP as the energy to drive things through the nucleus, and this stuff is going to go through the transporter. That purple barrel. Okay. So this is a level of control. Remember, diffusion is based on concentration. Active transport, you can control what is going in and out. And so this is really important for larger proteins, your RNA molecules, such as messenger RNA and tRNA, and your ribosomal subunits. Okay. So through the nuclear pore, small things less than 9 nanometers can go through by simple diffusion across the membrane, and larger molecules up to about 26 nanometers go through by active transport. So I want you to think of some of the things you learned about in general biology that would be transported across the nuclear membrane. Okay. So we've talked about messenger RNA, right? That's your gene expression RNA molecules. Your tRNAs, transfer RNAs, also important in translation. Your rRNAs are ribosomal RNAs. And we'll talk about this in a minute, but these are actually assembled in the nucleolus into ribosome subunits. Oops. Subunits. Okay, so the ribosomal subunits, these things down here, are actually what gets transported out. Those are all important for protein synthesis. And then some proteins that are needed for transcription and for DNA replication. So you know about these proteins like RNA polymerase and DNA polymerase, polymerase, and all the proteins that you learned about for DNA replication. Those are made in the cytosol and then have to be transported back into the nucleus for them to do their work. And this is showing you these little groups of 4. It's actually groups of 8, but I can't draw it 3D. Those are your histones, and the light blue is the DNA wrapped around it. An important feature of proteins that are going to be destined to go into the nucleus is a nuclear localization signal. Okay, so this is amino acids. Let me write that out. Amino acids. And it's a sequence required for proteins to be imported. Import. Why can I not think of that? Export, import. Imported. Into the nucleus. And it's interesting, the nuclear localization signals can range in size between 80 to 30 amino acids. Some are what we call monopartite, which means one chunk of sequence. Some are bipartite, means 2 regions required. But what I want to show you here is that here's the NLS for this specific protein. And when you mutate it, or you change that sequence, you can lose the function. So now it no longer functions as a nuclear localization signal, and now the protein is outside in the cytosol, not in the nucleus. Reverse of this is nuclear export signals, right, and NES. So there are signals, again, amino acid sequence. You can see all these leucines, and they're required. Sorry, let me just look at something really quick. Let me see if I can pause this for a second. Oops, no, never mind. Don't worry about it. Sorry, hold on. I just want to check something on my other computer. I don't want to say something incorrectly. Of course, my computer's slow. So it's amino acid sequence, and what they did here is they had this flip protein that normally localizes to the nucleus, and they cloned in on it. They cloned in a nuclear export signal, and so now the protein leaves the nucleus. Excuse me. So again, you can manipulate genetically at the DNA level, so using recombinant DNA, you can change these DNA sequences, which you know would change the RNA sequence, which would, which would change the amino acid sequence. And so you can manipulate these sequences to figure out, is this a true nuclear export sequence, or is this a true nuclear localization signal? And you can add these sequences onto other proteins to make them move into or out of the nucleus. Keep this in mind because you have a homework question that asks you about doing this. So let's talk about the two techniques from these two chapters. Excuse me. One is making a fusion protein. Okay, so in this case, we're using green fluorescent protein, GFP. And what you're doing, again, at the level of DNA, is you're adding the DNA gene sequence of GFP to whatever protein you're interested in, so that when it is transcribed and translated, it makes a protein that basically has two proteins stuck together, so they're fused. And so you can follow, by looking at the green fluorescence, where this protein is going in the cell. So this is just an example. Bright field, remember, is just our regular microscopy, and this is looking at the green fluorescent protein signal for different proteins that they're looking at. Okay. The beauty of this is you can look at live cells. Okay. So a few chapters back, we talked about immunofluorescence, where you're using an antibody to tag a protein to see where it is in the cell. But to do that, you have to fix and kill the cells so that you can get all those antibodies and material into the cell. With fusion proteins, you can look at live cells. And the very cool thing is, they've been able to manipulate this green fluorescent protein sequence so that it glows different colors, so that when you use your fluorescent microscope, you could have some that are looking red and some that are looking blue and some that are looking green. You got pink. So in theory, you could make multiple fusion proteins, let them be expressed in your cell, and have these beautiful mixes of colors. So remember this technique as a way to look at proteins, maybe for your homework assignment. Hint, hint. All right. Fluorescence in situ hybridization, also known as FISH. This is very different. Here we are looking at nucleic acid. Okay. Okay. Tagging. Okay, so let's explain this. So we've been talking a lot about using antibodies to detect different proteins. What if you're interested in a specific DNA or RNA sequence? What you do is you make a complementary sequence, and we call this a probe, and you label it with a fluorescent dye. Okay, so you could imagine it's got some fluorescence on it, and when you put it into the cell, it will hybridize or base pair with the complementary sequence. And you know A and T and G and C, right? So what you can do is look at, say, chromosomes and look to see if a chromosome has a specific DNA sequence on it using this fluorescently labeled probe. Okay? So we're not talking proteins anymore, we're talking nucleic acid. This is really cool stuff for genetics, and we will be coming back to this technique for our cancer biology at the very end of the semester. So I just wanted to show you a few pictures of some FISH experiments. So here they're tagging genes that are specific to chromosome 15 or chromosome 17, and you can see the different colors lighting up. Okay, so this is one way to look at, do these chromosomes have this specific gene sequence? Are they missing the sequence? Perhaps like in a cancer cell? This is just expanding or telling you some of the things that we can do is look at the telomeres. That helps us just look at all of the chromosomes. The centromeres, you remember, this is where the spindles attach to separate chromosomes in mitosis and meiosis. Chromosome painting, we will look at an image of this. This is what they did for the chromosomal territories. Okay. And what's really cool is the gene-specific probes. And you can actually use this to find RNA, like mRNA, in a cell also. Okay. Because of the way you have to do this, these cells would be dead. Okay, in order to get the probes in there, you have to denature some of the DNA. But again, you can see different colors are used to light up. Here in a genetic example, they're showing you that normally people have they have 2 chromosome 13s in green and 2 chromosome 21s in red. And this cell has trisomy, it has 3 chromosome 21s. So this would be a cell from a person with Down syndrome. All right, so you've got some tools now to be able to look at proteins and where they're localized and where they go. So let's talk back to the nucleus and about things moving in and out of the nucleus. Okay. So RAND-dependent import. So there's lots of details. This is a figure from your book, and the book goes through nice step by step. I'm only going to highlight a couple things here. So this could be a protein, cargo protein, maybe DNA polymerase or RNA polymerase or some protein that's needed in the nucleus for replication or transcription. And so they're going to have a nuclear localization signal, right? They bind to a protein called importin. So import, oh, there's my M. I don't know. It's very weird to lecture in your room with nobody listening but yourself. So that's why it sounds kind of funny once in a while. Okay, anyways, they bind together and they're going through that transporter in the nuclear pore complex. Okay, so importin takes it in, and then there's this protein called RAN, shown in green. And RAN is the protein that's actually providing the energy to make all of this happen. And so you can see RAN is bound to GTP. So sometimes our cell uses GTP for energy instead of ATP. So RAN interacts with this importin and cargo protein. RAN binds importin, releases, say, DNA polymerase into the nucleus. RAN is then transported out, and what's important here is, remember, if you're going GTP or ATP to GDP or ADP, this is called hydrolysis. Okay, so you're losing, I hate when it does that, you're losing a phosphate, right? And so that burst of energy release allows importin to be released, and it can go bind another cargo protein, and RAN to now be also ready to go back in to the nucleus. And I'll show you that RAN pathway in just a minute. So RAN-dependent import means you're using RAN, you're using GTP energy to move stuff through, stuff through the nuclear pore into the nucleus. All right, the next few slides are flipped. So the nucleus is down here, the cytoplasm is up here. I tried to flip it around so we were all moving the same way, but it's kind of hard to read upside down. Okay, messenger RNA needs to leave the nucleus so that it can go into the cytoplasm and it can attach to ribosomes and it can be translated. Messenger RNA leaves by what we call a RAN-independent pathway, okay, which means it has other proteins that bind to the mRNA and take it through that nuclear pore complex. But what I want you to see here is that it still requires energy. So remember, these are all examples of active transport. Okay. So ATP hydrolysis, and you don't need to know these different proteins here. ATP hydrolysis is the energy to move the mRNA out of the nucleus into the cytoplasm. These proteins are then recycled and goes around and around. Others, such as tRNA or ribosomal subunits, leave by a RAN-dependent pathway. So again, nucleus down here, cytoplasm up here. RAN GTP is in blue. It binds exportin, okay, which is binding a protein with a nuclear export signal, which could also be binding to, let's see if I can draw a, oh my goodness, hold on, my brain just stopped. This is my tRNA, okay, so the RNA molecules bind a protein. The protein has the nuclear export signal. It binds the protein exportin. exportin is binding RAN-GTP, which if you look back here, you got RAN-GTP in the nucleus, right? Takes it out through the complex. Again, you have GTP hydrolysis. We release that energy, the complex breaks up, your tRNA could go off and find a ribosome, everything goes back into the nucleus, and your RAN-GDP becomes RAN-GTP again. Okay? Exactly the same thing that's happening up here. Just remember we're upside down in these other pictures. So, GTP, ATP, used as energy for active transport, can be RAN dependent, it can be RAN independent. And so a couple things to remember about the RAN cycle, right, is that you have a couple important proteins. GEF stands for guanine nucleotide exchange factor. Okay? And so it is making RAN GTP in the nucleus. In the cytoplasm, so this is our nuclear membrane and our pores. Okay. In the cytoplasm, you have something called GAP, right, which is GTPase protein, which means it hydrolyzes GTP to GDP, releasing that energy. And then the last protein to note here, NTF2, which is nuclear transport factor 2. Factor 2, sorry. Factor 2. This guy binds RAN-GTP and shuttles it back into the nucleus. So, did I say GTP or GDP? Okay, it binds RAN-GDP. So remember, RAN-GTP is used to move importin back to the cytoplasm to grab another protein. It's used to move, and you can see right here, importin. It's used to move exportin, right? And then you've got to get the RAN-GDP back into the cytoplasm to make more RAN-GTP so you can continue this pathway. All right, so what else is happening in the nucleus? You've got this cool thing called the nuclear lamina. And lamin is a type of protein. It's called an intermediate filament. And we are going to come back and talk about this again in Unit 3 with the cytoskeleton. But just understand that the nucleus has got its own nucleoskeleton. Right underneath the inner membrane. So what's really cool is you can actually dissolve using detergents, and you now know that detergents break up membranes because we did that to isolate proteins from our membranes. So you can dissolve the nuclear envelope with some detergent and you will still see something in the shape of a nucleus, because these are all very stable proteins that make up this meshwork that give the nucleus its structure and integrity, and it gives it strength so that it can protect all your DNA in there. Really, really cool stuff. And what also is interesting is that lamin is a protein that's not found in plants or fungi, so only animal type eukaryotic cells. What else is happening in the nucleus? Don't forget transcription. So if it's been a while since you've thought about gene expression, review this slide. So remember, transcription is DNA to RNA. Your pre-messenger RNA gets processing, which means it gets a cap, a 5' and a 3' poly A tail, and the introns are spliced out, and we get a mature mRNA, and that is what's going to be exported to the cytoplasm for translation. What else is happening in the nucleus? Well, we have, our nucleolus. That, remember, the function of the nucleolus is to assemble ribosomal subunits. And this is really important. Subunits. Okay. The ribosome does not get put together until it's in the cytoplasm and it finds mRNA. Okay. But a ribosome subunit is made up of ribosomal RNA plus protein. Okay, so these RPs are ribosome proteins. You've got your ribosomal RNAs. They come together to form the large and the small subunits. Those large and small subunits are the ones that are transported. So there's no ribosomes in here. Okay. Remember that the nucleolus is an area, okay, that this is happening. Okay, and we're not going to worry about distinguishing the granules and the fibrils. All I want you to know is that there's this area where you've got ribosomal RNA being made and ribosomal subunits, RNA and protein, being put together for export. All right. What else is happening in the nucleus? Okay, so I like this picture because here's our envelope and a pore. And this nice pretty pink is your lamina, your nucleoskeleton, sorry. Okay. And then you've got your chromatin. And don't worry about these early and late replicating domains. But you've got your DNA and their proteins. And they're somewhat organized inside the nucleus. So there's locations where you can find the different chromosomes, and they're actually attached to the lamina. Let's look at this a little further. So your book talks about chromosomal territories. This is fish. This is chromosomal or chromosome painting. Okay, so they've painted the different chromosomes different colors, right? And remember that your chromosomes are all like this. They're only like this, like the little X's or the little fat sausages, during DNA replication and mitosis or meiosis. But normally there are these squiggly things like this, right, throughout the nucleus. And the chromosomal territories just means that each chromosome seems to be organized into a certain region. I want you to understand that every time you looked at a cell, the chromosomal territories could be different. And we think this has to do with gene expression. So it's not a fixed location. You're not always going to find chromosome 2 right here. But when you are looking, there is distinct territory. So it's not like this with all the colors, right? They've got their own regions. And so this is just another picture showing a chromosome territory. And what's really happening down here is you've got the lamina, maybe here and here, And you've got lots of chromosome here, and then little areas that pop up, and you can see we talk about transcription. So a lot of our chromosome is tightly wound up in what we call heterochromatin, and you can't do transcription. This part is called the euchromatin, and that's where the chromatin opens up and transcription can happen. You can make messenger RNA. And again, so we think there's this organization of the chromosomes and the DNA to keep it from getting tangled up, right? So again, from keeping it from that ball of yarn that gets all knotted up and keeping it organized. Okay. I think I've talked about this twice now. Important, gene expression, right? So DNA, RNA to protein on the ribosomes in the cytoplasm. All right. This is translation. Making a protein. Proteins are made of amino acids. We're going to come back and talk about proteins being made on the ER. The other thing I want you to remember, and we talked a little bit about this last chapter, is that proteins can be modified. So we talked about co-translational modifications, such as N or O-linked glycosylation that happen in the ER while the protein is being made. Post-translational modifications, remember, happen in the Golgi. And honestly, I don't know cis, medial, trans, so we'll just say Golgi in general. And there are things like hydroxylation and sulfation and methylation and phosphorylation. So all these different types of functional units can be put onto a protein to help give it function. And if it happens after translation is done, it's called a post-translational modification. So you can add functional groups. Oh, my handwriting is horrible tonight. You can combine subunits. So that means each of these different colors here in this diagram represent an individual protein. When we put them together, we have hemoglobin. Okay, and remember this is called quaternary. Quaternary. I can never spell. You guys have realized that by now. Structure, quaternary, sorry. I think this is right, quaternary. Anyways, you know what I'm talking about. 4th degree structure, right? See these heme groups? This is what the book is referring to as prosthetic. So these are helper groups, or helper molecules, I should say, that are important for protein function. So heme is what binds your oxygen, so that hemoglobin can bring oxygen to your cells. Other types of post-translational modifications can involve actually cutting the protein. So inteins and exteins, remember introns and exons are in RNA molecules. Inteins and exteins are in proteins. So some proteins can self-splice. So they can actually cut a piece of the protein out and rejoin it. So the inteins go out and you're left with the exteins or the functional protein. Some proteins, like insulin over here, are processed by other proteases, which means, let's see, processed by a protease. Protease is an enzyme that cleaves proteins. So in this case, insulin is made as this big single chain of amino acids, and then this orange part and this very end terminal part is cleaved out, and the insulin functional protein is made up of two chains. Came from an original single peptide, but it's not functional that way. Whoops. Disulfide bridges between cysteine amino acids can connect these two chains. Disulfide bridges sometimes are made co-translationally and sometimes made post-translationally. And another post-translational modification could be the removal of a signal peptide. So we're talking a lot about signal sequences that direct the proteins where to go. Sometimes these are cleaved co-translational, sometimes they're cleaved post-translational. Okay? All right. We got a few more. Protein targeting and sorting.